Ladies and gentlemen, distinguished colleagues, now we begin with the working part of the conference. The first plenary lecture named All the New Breeding Goals and Challenges will be held by Dr. Felicity VEAR. Today I am very honored that I am sitting in between Dr. Felicity VEAR and academician Dragan Škorić, who was the chairman of the several times mentioned conference in 1988. So please welcome academician Škorić for all they have done to our sunflower community. After Dr. Felicity VEAR is certainly one of the brightest stars of our sunflower galaxy. After a studentship at Indra Clermont-Ferrand, Felicity VEAR obtained her PhD from Reading University, United Kingdom, downy medio-resistant and male sterility in sunflower in 1973, and then joined Indra as research scientist working at first with Patrick Leclerc. She was research manager of the genetics and breeding team at Indra Clermont-Ferrand, France, 1986 to 2015. Since retirement, she is an honorary scientist attached to the sunflower genetics team at Indra, Toulouse, France. Research subjects, disease resistance. Resistant to downy mildew since the first attacks in France in 1969, testing methods, genetics of major gene resistance, non-race specific resistance, resistance to Sclerotinia and Phomopsis, testing methods, heredity of resistance, and identification of QTLs. Breeding methodology and related subjects, recurrent selections, methods for testing, combining ability, breeding of disease-resistant inbred lines, organization and the rationalization of maintenance of cultivated genetic resources, heredity of earliness at flowering and harvest, effect of cytoplasmatic male sterility, genetics of pollen and nectar producing, and many more. She was a member of the International Sunflower Association Board from 1992 to 2017, then ICA President from 1996 to 2000. For her great and world-recognized contribution to the improvement of sunflower production, she was awarded a Pustavoj Medal in 2004. I had the privilege of collaborating with Dr. VEAR on the ISA board. Her experience and support were of invaluable help to me, and I can say that we are all here today thanks to the trust she has shown to the Novi Sad Institute and to me personally. Dr. VEAR, you can now begin your presentation.
Thank you very much, Dr. Miklic. It's nice to see everybody here, and I'd like to thank the organising committee for inviting me to do this introduction to the conference. So, all the new goals and problems in research. I just was going to talk a little bit, it's a bit of history about variety type, and then in breeding goals I divided it up into the four lines you can see. Some of them you probably won't agree with when you go into detail, that something is perhaps not cyclic but continuous, and what I say aren't now priority. Some people may well say that they are priority, but that's the discussion of the conference. The important things, I think, are to know why certain goals are more important or less important, and for a few questions, how they've changed, but mostly that's what we'll hear about during the next 3 days. So just variety type, just shortly, this photograph has more than 50 years old. It was taken, I think, in 1968, with the very first hybrid in RAR 6501 compared with Peudovic. And when I first joined the Sunflower team, it was a question of uniformity that was most important. And the great change, because perhaps not here, and I'm preaching to the converted, but in Western Europe you hear whether shouldn't we go back to population of open pollinated varieties because they'd be more adaptable. But when you see that hybrids have depended on selection for self-fertility of inbred lines and hybrids, it would be very difficult to go back again. And just two other points. For breeders, it's the possibility of rapid innovation. And for public research, I think that having lots of inbred lines makes it much more easy, the maintenance of genetic resources, rather than having to multiply open pollinated varieties. Now, if we go on to different goals, the first one I put are goals that are now less important because breeding has been successful. Why? Because breeding has been successful. And the first one, I think, is CMS. You could say we were lucky that the Helianthus speciularis that Heiser provided to Leclerc gave us a CMS that has caused no problems. At the time, there was the problem with maize Texas cytoplasm susceptible to a Helminthosporium maydis, but with sunflower there hasn't been a problem. And since then, there have been possibly 72 different CMS reported. Probably there are quite a lot that are the same, but there's certainly plenty. The restore genes are good, and in addition, recent work has shown that the pet cytoplasm may well be more drought resistant than the ordinary annual cytoplasm, so it may be a plus already. The second goal that is now, probably most people don't even know about, is the question of seed drop. When we first started, probably remaining from wild annuus, there were problems when hybrids or lines were below 15% humidity. There was seed drop. We went round banging on the heads to check whether the seed dropped before harvest. Well, it's been eliminated now just by selection for breeding for yield, but it's something, if people are using annuus, it's something they should think that could occur that they need to select against. In particular, just a detail, I don't know whether many people know the line now, but it was in particular a problem with the American line HA61, from where PL2 and some sclerotinia resistance and branching appeared that we had problems with, when it was actually used as the male parent of a genic hybrid. Other goals where breeding's been successful, I would say oil content. This was a comparison of varieties in the different periods from 1970 to 2010, and there's been some increase, but in fact the first, I don't know, I can show you, yeah, oh no, sorry. Well, anyhow, I'll indicate the first variety, rather over 105.
This one? This one? This is in the middle?
Here.
No, below. Here.
Yeah, this one here, above 105, is in fact peridobic, so that you could say that in fact Pustervoit did most of the work, that we haven't really changed. I mean, there's a bit of increase up to 108, but there's not been a great change, and it's something that's got to be maintained, but probably oil content of sunflowers won't go much higher. And the other goal that's been successful is high oleic. From Solidopsis mutation, now yields are approximately the same as those of conventional varieties, and there's high and stable oleic acid. The question here, I would say, is that it's rather a paradox that there's been, especially in the US, there was a lot of work or question to have as low a saturate as possible. And in fact, the new sunflower types, high oleic and now high stearic, have gone in the opposite direction. They're less unsaturated. Perhaps it's because this is the desaturases that don't work anymore, and it's easier to knock things out than to find something new that works. One wonders whether you could have sunflowers with high in omega-3, but here you'd need a new desaturase, and perhaps it's more difficult. The other subject where breeding has been successful, and it was first known here, I mean, it's the right place to talk about it, it was where here in Novi Sad that Phomopsis was first identified and first breeding started. And now I'm just showing here a graph to show results in France, that now most of the varieties grown in France have good resistance, and there are very few that have poor resistance. So it's something that needs keeping, but is no longer so much a subject, at least in Europe. In the US it may be more a problem, and this is, I think, quantitative resistance. It's something which should last. So it's, I think, a successful achievement of breeding. Gold's less important, that I would say, because of agricultural climate changes. Here it's disease resistance. When we first started in the 70s and 80s, botrytis was a problem, but it disappeared. I don't know whether because sunflowers dried down more quickly, because of genetic changes. Already in the 90s, I think botrytis has disappeared. And for sclerotinia, which is something I've worked on a lot, and there's been an enormous amount of work, not only in France, but in Argentina and the US. I hope some of the less importance of sclerotinia is due to the breeding effort, the research we did, but I think it's also partly due to where sunflowers are grown. They were pushed out of the irrigated zones in favour of soybean or maize, and with climate change, I mean, now with the hot temperatures, it's not very favourable for sclerotinia head rot. But if sunflowers move further north or are sown earlier, this could come back, I don't know. And the third disease I will mention is FOMA, where we were very worried. It was very worrying in the beginning of 2010. It's now less talked about. You could say whether it's agricultural people grow varieties that are not particularly susceptible, or it's been eliminated in breeding, but it's another subject which appears to be less important now. If we go on to goals that are always important, of course, it's seed yield. And there the main point is how to get through gains, genetic gains in yield, through to the farmers. And here, I'll do it here. The first graph is national yields don't increase very much, although in fact in 2021 the national yield in France went up pretty well as far as the green line. The green line is large-scale trials, and this was the other graph was the small plot trials that we did in 2001, which showed the increase in potential yield from Berry the Big, which was the first variety, up to varieties that showed 140%. And there are at present there's a new research program which has been carried out to see what's been happening more recently. So the question is how to get there is the genetic gain, how to get it through to the farmers. Here I just mention a few new tools because the subject remains the same. You could start with now genomics and genomic selection. Here the first work in Toulouse showed that oil content, that genomic selection could be quite accurate, whereas yield is much lower, although in this work it was the same as flowering or height. What I do wonder is whether, since it's such a competitive subject, whether it'll be published when firms manage to get good genomic selection for yield. I don't know whether they'll publish it. It would be nice to know whether how it's increasing. The other important points about yield are now automatic phenotyping, which makes it possible to analyze physiological characters, to make large-scale measurements, to be able to divide up yield into the characters which decide that. And then the third point is to make, by models, make most use of a small number of trials, rather than having to do lots of trials in all sorts of conditions, to take into account environmental conditions and management. So this should help to reduce the cost of breeding for yield if fewer trials are necessary. And on yield, I've just finished by comparing what we did in 2005, 2006, when we started to work with the agronomists. And this was when, measured by hand, what we called residual leaf area, which was the green leaf area 3 weeks after flowering. And it was shown that compared with height to flowering date, total leaf area, and various other characters, this was the best correlated with yield. And I found in publication by Philippe VEAR that in fact the leaf area duration, which is the same thing really as residual leaf area, measured by satellite reflectance, also gives the same results. So it's nice to have a confirmation that measures by hand and measures by aerial photography confirm the same results. I go on, goals always important. I'd say drought resistance. This is because when I first started, I say since there was a spread from the chilodendrons in Eastern Europe, perhaps in Eastern Europe you would say it's always been a question, I don't know, but I can remember lots of people were doing crosses with Helianthus argophilus. The advantage is that it crosses quite well with sunflower. But I would say that I've never seen a hairy sunflower. I don't think that in fact it got through into breeding genes from Helianthus argophilus. And now in cultivated sunflower there are much more improved measurements, and genomic work will help to identify the physiological characters within, it may be wild sunflower, but in cultivated sunflower there are quite a lot of differences. And the question will be whether it will be real resistance or whether it will be escape from drought by sowing at a different date. If you go on to goals, there are a lot of goals what I say are cyclic importance. Take the example for protein content halability. This has been, each time there's been a problem with the availability of soybean. It started in the 80s, and then there was another series in the 90s to find a replacement for soya. I would say in the past it was proteins for animal feed, but nowadays it may be also for human food. There are confectionery sunflower, but I wonder what would be the use of what I call serious proteins, perhaps in the form of flour, and what it would be possible to do, because there are frequent negative correlations between protein content and oil content, and positive correlations between hullability and percentage hull. And what could be done to have a high protein sunflower? Would it be possible to have different varieties from oil sunflowers with high protein, but will it be possible to have hullability with thin hulls, because it's rather a waste to spend the energy of sunflower on hulls rather than the kernels. The other goals of cyclic, what I put in cyclic importance, I don't know whether it's a subject I know less about, perhaps people will say it all the time. I can remember many years ago that the work at NCSet at Toulouse, they were making biodegradable plant pots from sunflower stems or heads. And now it seems to be quite important, it's to have biomaterials to replace plastic. And this is a sort of secondary harvest, so it would be good for the farmers. But I would say how much can be removed from the field, how much organic matter should be left, how much of the stems should be left, and whether, since it's a rather secondary character at the moment, should they be breeding. Going on to cyclic importance, Well, I started off with the disease I know best, downy mildew, and then there's orobanche. And in fact, when you think about it, it's cyclic importance. It's because the pathogen has races, and when there's no new races, things calm down a bit. People go on breeding, but there's less need for research. But one wonders now whether they won't become permanent subjects. So just to compare the two, where now downy mildew is pretty well worldwide, and appeared in the US at the same origin as Helianthus annuus. Orobanche is not found at present, as I understand right, in the Americas. It's more recent, and it'd be interesting to know why. For downy mildew, there are lots of resistance genes, where there's about 35 at present, found in wild Helianthus annuus. And the thing I wonder about is, well, they're all really different. I go back to PL6 and PL7, which came from wild annuus and from, I can't remember which, but anyhow, another species, that were both knocked out by the race that we called 304. They both were knocked out at the same time, so one can wonder whether, for the pathogen, they were the same gene. And it may be that among our 35 genes that we've got available, it may be the same thing. We distinguish them by their origin, or by their molecular structure, or by their markers, but in fact there may be some that the pathogen thinks are the same thing. Compared with that orobanche, there appear to be fewer resistance genes, and there are even some lacking for the most virulent races that appear. Well, for downy mildew, there are differentials, whereas differentials are still needed for orobanche. Going on, I would say there, orobanche, recent work, the resistance mechanisms have been well identified, the different stages in the disease progression, whereas it's less well known for downy mildew. We know the symptoms that appear, but the details have been less studied. And for orobanche, the resistance gene has been sequenced, but I don't think it's the case for a PL gene. But on the other hand, now for downy mildew, races have been distinguished by genomic polymorphisms, whereas this is not yet the case for orobanche. My questions are, will it make it possible? It would be very useful if, when you've got symptoms in the field, you could just analyze molecular and not have to do a whole lot of biotests with differential lines. And then the day when resistance of genes have been sequenced, will it be possible to select the strongest, the ones that are most different or most likely to last? Going on to other diseases, here I would say verticillium, of cyclic importance in France, because I can remember in the 80s we had this disease, yellow spots on the leaves, verticillium in the valley bottoms where we had macrophomina on the slopes and hilltops. And since 2010 it's come back, and I don't know why, and why there's more in Argentina than USA. Is it a question of climate? Is it a question of sunflower genotypes that are grown? And I think there still remains the question of, are the races, in the sense that we know them, for downy mildew or orobanche? And one quite original response is not only by sunflower resistance genes, but they could be controlled by biofumigation with glucosinolates from ground up mustard leaves. So there's quite a lot of interesting questions for verticillium. Other goals of cyclic importance, pollinators and bees. Here the first work was done when hybrids were developed. We had to find out how far bees moved, how many bees you needed to have good hybrid production. And well, it has been shown, even now that sunflowers have been hybrid lines and hybrids have been selected for self-fertility, that the presence of pollinators gives improved yield. So it's still important to have bees or other pollinators on sunflower crops. And now the question is more of environment. For the beekeepers, it's a question of honey production, and in general it's to maintain or increase or favorize pollinators, not only for sunflower but for other crops. And there have been quite a lot of recent work. Here I just show that corolla length in the US they showed that the shorter the corolla, the more bees visited. Bee attractivity also appears to be related to nectar volume, but this may be a problem in question of drought because nectar volume is reduced when plants suffer from drought. And there's a third point which we haven't really got the answer yet, which is being studied, that we found already in the 90s, that there are most of sunflower lines have the nectar is made up of fructose and glucose, and only a few have sucrose. And the question, bees appear to like sucrose, but whether it's a good thing to have them remains open to question. I can remember comparing the hybrid 894, one of the first in the 80s. Neither of the two lines had any sucrose, whereas another hybrid, a French hybrid, there was one parent which had sucrose and the others didn't. And in fact, the hybrid production for the variety that didn't have any at all was much better than they must have. The bees must have stayed on the line which had sucrose. But this question is still under study to know what to recommend to breeders if they take this into account. If I go on to new and recent goals, herbicide resistance you may regard not as recent, but for me it's recent, since about 2000. And I would say this has been very successful for breeders and much appreciated by farmers. But the question is whether this will be long-term, with the question of reducing use of pesticides and whether this can continue to be used, or how long this can continue to be used, and whether there are any other possibilities. And the question is how much resistant will be developed by weeds, and how important will organic sunflowers become. In France it's about 10% in 2021. And are there other ways to reduce the weed problems? Rapid seedling growth to cover soil, or what are called mixed cropping. Now it's done where you have a different crop to cover while the plants, while the seedlings are growing. And going back to mechanical harrowing, and my question is, can breeding provide any new answers that do not require pesticides? The other second new recent goals, this is because it's now announced it's a problem that's always existed, especially for small areas of sunflower. And now with the encouragement to have more hedges and more trees, and to, as birds have reduced, but we try and re-increase them, it will carry on being a problem. It's both at emergence and at harvest. And I think this will be one of the most difficult topics to have success. Well, would regular germination and fast seedling growth help? Would rapid dry down help at harvest? I think that this is a question it's necessary to undertake, and I hope there are methodologies now to do better than what we did 20 or 30 years ago. And the other question of new and recent goals, which are announced, which have always rather existed, but now there's research, are cold resistance and seedling vigor. You could say that we worked on seedling vigor. We used to note the seedlings which appeared the most vigorous in trials or in red lines, but there again it was difficult. I can remember the line that I think people still know of, HA89, which was widely used in the 80s and 90s, It was very yellow, and if you were deciding which plants to keep, you wouldn't have kept that when it was a seedling. It was perfectly all right when it got flowering, in fact very vigorous and good. So we said that it was because it was growing roots, but whether that was true or not, I don't know. So that now, if somebody can know something about the importance of seedling vigor, it would be very useful. In the 90s, we did some winter crop trials in Morocco and Spain, sown in November, without frost, and these did quite well. There was some sclerotinia in April when the plants were flowering, but how much this could be spread. And now the question is, would it be better to have early sowing to escape from drought? And so whether it's a resistance to cold or whether it's rapid and uniform growth under low temperatures, this could help to reduce weeds, could help to reduce damage. In work, there was a thesis in Toulouse, that showed genetic differences for chlorophyll content, osmotic potential, base temperature of germination, and hypocotyl elongation. Here, I don't know whether you can see it very well, but there are two varieties that have different base temperatures and different rates of hypocotyl elongation according to temperature. So this is something which there must be genetic variability. This is something which could be done. And in general, you would say a better knowledge of the physiology of germination and seedling growth could help several breeding goals. The last new and recent goal, you could say it isn't really, it depends what you're talking about, is possible changes in idiotype. In the 80s, there was a lot of work on dwarfs. Here, this was the mutant dwarfs that Leclerc obtained, but in fact, whatever their origin, whether they were from cultivated sunflower or wild sunflower or mutations, they gave spore seed. They looked very well, the plants, but they gave spore seed sets, so that stopped off. There was also the idea when Montpellier in France, they made crosses with Helianthus mollis, which has sessile leaves. That was the idea that you could have higher density if leaves are sessile, but I don't think anything came of that. And more recently, one hears of, should you go back to branching? It's a bit like populations. Would it give more adaptability according to conditions? And here I would say I was looking for a good photograph of branching, couldn't find one. I could only find ornamentals. So it appears to me that I'm not sure that other than use as biomass or intercropping and for ornamentals, branching would be very useful. And if you had a sunflower like that, it would take a lot of breeding. I do wonder, I'm not a specialist on wild Helianthus species. Are there any that have a structure that would be interesting to have a sort of rosette type that then produced several stems? I don't know whether that exists, and certainly it would require a large breeding effort. So, in conclusion, most goals are in fact long-term or cyclic: yield, drought and disease resistance, insect pollinator activity, and the relative importance of oil and protein. You could say CMS and oil quantity and quality have been quite successful. And the new characters are not really new, but before it was difficult to obtain results: cold resistance, seedling and young plant growth, and bird resistance. It's not real, I don't know, you could have real resistance. And I wonder whether the new tools, models, aerial photography, phenotyping, genomics will make it possible to obtain significant advances in breeding concerning these goals. I think that bird resistance will probably be the most difficult. And to finish, I'd like to thank all my colleagues at INRAE Toulouse, both the geneticists and agronomists, and at Terry Novia. I'd like to thank Promosol, which an organisation where we collaborated with all the breeding firms in France, and it's now a new organisation called Celiopro. and the colleagues in all these firms, which are many of whom I see here, and also ESA for their comments on my talk, for their help, for use of their figures in this presentation, and especially for making it possible for me to follow progress in sunflower research in the last 6 years. So thank you very much, everybody.